Health experts warn that we're overusing the drugs we count on to fight infections. Antibiotics have saved or improved the lives of millions of people. But as bacteria become more resistant to those drugs, it's getting harder to find new ones. One of the new places where scientists are just starting to look may surprise you. Ants. Rarely seen in the wild, mysterious and misunderstood. Where did the Gila monster get the special traits that keep it alive in our dry, harsh deserts? Well, the answers probably lie in decoding the Gila monster's genes. We'll show you how researchers are doing just that and the surprises they're finding. And speaking of lizards, you may know that some of them can actually regenerate their tails. How? And could understanding that question hold out hope that one day humans might be able to regenerate a missing limb? Hello, I'm Professor Vanessa Ruiz, and you're watching Catalyst. This time, animal superpowers. This YouTube edition of Catalyst is brought to you by Arizona PBS and Arizona State University. Life has problems. Science turns them into questions that can lead to solutions and even innovations. This is Catalyst, shaping the future through science research at Arizona State University. This is the garden ant, one of the big bugs here at the Phoenix Zoo. Obviously, these models are thousands of times bigger than actual size. Now, one of the few things that humans and insects do have in common is diseases. And scientists are digging deep to figure out exactly how some insects, especially ants, are creating their very own antibiotics.
Ants are very successful. If you go out into most ecosystems, over half of the insects you're likely to find will be ants. They're really, really abundant. On top of that, they're also diverse, so there's somewhere around 15,000 species of ants, and they've been successful on the planet now for somewhere around 150 million years. We think that most of the species have found ways to effectively deal with pathogens, but just like humans, new diseases arise and new colonies can get hit harder than others, and so that's probably their number one threat is disease. Ants live in these large colonies, they have a high interaction rate. It's just like humans in cities where that's where illness spreads. When someone gets a cold and they sneeze on a train, they can send that cold to hundreds if not thousands of others. And ants deal with the same problem, compared to something like a solitary insect like a grasshopper or a beetle, which doesn't live in this colony. They don't necessarily have the same pressures from disease as ants might have.
And just like humans, ants need a little help fighting off an infection.
The major goal of our study was to figure out if we can narrow down what species would be the most interesting to look at for future antibiotics. There's more different species of ants that live in Arizona than any other state. So this is an ideal location to continue our studies and look for other species that might produce antibiotic compounds.
First, Dr. Pennick needs to collect some ants to study, but he doesn't always need to go very far.
All right, so right here is actually an ant colony. And so what we do for this study is we just walk up and down the streets, and because ants are so common, it's pretty easy to find a range of species. And so right here, you can tell there's a little piece of a nest, a part of the mound, and then the ants are coming out, they're digging right now. And so this species, let me see. Yeah, these are actually crazy ants is what they're called. And so we actually study these, and these do produce some antimicrobials. And so what I'll do, I use this device, it's just called an aspirator, and it's essentially a mouth vacuum that you use to suck up ants, and they go through this nozzle into this container, and not in your mouth, thankfully, because we have a screen. So let's see here.
Dr. Pennick then takes the ants back to his lab.
In our research, we have to keep ants alive in the lab, and so we learn a lot about animal husbandry. How do you take care of an ant colony? What's an ant's favorite food? Each species has their sort of own requirements. And you start to notice some of them do get sick. They get mostly fungal pathogens. Some of it could be bacterial, but you'll start to see ants pile up in the corner. The ants are very meticulous about their colony. So if there is an ant that dies, they quickly find that worker and they'll drag her outside and put her in a containment trash area. Maybe we could call it a graveyard. Ants have all these behaviors that they use to protect themselves from disease once they get in their colony. One of the challenges with studying antibiotics that ants produce is that ants are really, really small. And so, if you're trying to extract an antibiotic or a medical compound from a plant, it's usually pretty easy. They have big leaves, you can get a lot of material, but for ants, you just don't have a lot to work with. So what we did is we would take ants that we collected outside and we would soak them in alcohol overnight. And then we would take that alcohol off, we would dry that down and resuspend it in basically bacteria food. And then we would add Staphylococcus epidermidis, which is a bacterium that's common, it's on human skin. Probably all of us have it on our skin right now. It's not a pathogen, which makes it easy for us to work with. But if the ants could kill that with their compounds, then we knew that they were producing something that was antibacterial. And so we found ants that produce active compounds that do kill bacteria. And so what we've really accomplished here is the first step in finding a new antibiotic. But the questions are now, do they actually work against something that's resistant to our antibiotics that we have now? And it's a long process, so we have to narrow down first, are they effective? Then we'll have to look at identifying the compounds. And then you have to figure out actually how to make and synthesize them. But what is really fascinating to me is that these ants that sometimes are a nuisance, like fire ants, might actually help save our lives one day. And I think that is a pretty big deal. And especially thinking about these natural habitats that have higher biodiversity, places like the Sky Islands in Arizona or the tropics. These places are really holding a library of potential future drugs and medicines for us. And so I think there's a lot of reason to protect these habitats. And then there's also reason to look at even the lowly ant. They might save us one day.
There are a few of us, and yes, that includes me, who don't have the nerve to hold creatures like these. And you can find them here at the Phoenix Zoo. I'm talking about all kinds of reptiles. This is a Gila monster. It lives in the desert, and it can actually go for long stretches without water. It has venom, and yes, it can look a little scary. But the question is, how did the Gila monster evolve and survive with such strange traits? To get those answers, you've got to do the work to unravel its genome.
Biologist Melissa Wilson has a surprising reaction to venomous reptiles.
They evolved from ancestors that were in warm, wet, tropical environments, and they've managed to survive here in the desert. And I think that's wildly fascinating. What is it about them? Are there signatures in their genome that help us understand how you can survive in a harsh environment?
She's working with Gila monster expert Dale DiNardo to map the iconic lizard's DNA and how it evolved from prehistoric reptiles.
The name Gila monster is something given because of the impact of their bite, but in many ways they're not a monster by any means. They're really rather be left alone, and they spend a lot of time wandering the desert looking for nests of other vertebrates, bird eggs, small mammals, reptile eggs. In doing so, they got to travel a lot of distance over the desert, and they will have a home range that may be as large as 70 football fields. And they also have very bumpy skin, and each one of these bumps has a bone called an osteoderm inside of it to help protect the animal not only from predators, but from very common thorns of cacti and other plants in the desert.
Unlike many lizards, the Gila monster cannot regrow its tail, but its big back appendage does have another survival skill.
They lost that ability, and instead they store fat in their tails that allows them to survive in this really harsh environment.
They can go months and months without eating only several meals in a given year, and that'd be adequate not just to live, but actually to thrive and reproduce.
There's a peptide in their venom, which is actually just saliva, that is used now to treat type 2 diabetes. They're just wonderful critters.
So Melissa, by sequencing the genome, we can target and look at specific genes and see if they're upregulated compared to other lizards or other desert species, and really get a good understanding of what changes have occurred in them to enable them to do some of these things.
I'm mainly interested in sex differences, so they're sex chromosomes, so we can look at Gila monsters to study general features about how sex is determined across life.
In humans, that trait is inherited from the genes of our biological parents.
We are XX, XY sex determination, so that means it's the sperm producers who can make a sperm with an X or a Y, and that determines the sex of the offspring. In Gila monsters, it's swapped. So it's the females that make an egg that has a Z or a W, and it parallels our sex determination systems. I think it's really fascinating that you could look at a lizard and understand something about ourselves.
Genetic testing creates millions of data points for biologists to explore, but organizing and decoding the files is an enormous task.
So we look at the canonical DNA, RNA, protein, but it's now all in files. What's moreover is that you could, in the past, visualize all these on your own.
What is a DNA molecule?
And you could just open up the file and the sequence might be 100 A's, T's, G's, and C's, or even 1,000 A's, T's, G's, and C's. And it's a lot, and you could painstakingly go through. But now we're working on an RNA-seq file might have 40 million reads that are each 150 base pairs long. And then you'll have 6 of these files. So we're working in billions and trillions of reads, and it's not something you can do by hand anymore. And it's just going to get larger and larger.
Computer software can be extremely helpful in calculating genetic results and finding important patterns in the DNA sequences. Today, software coding is as useful in biological research as microscopes were in the past.
When we sequence a genome, we get a bunch of reads back. We get the little puzzle pieces. And some of them we trust more than others. And so we need to do a first pass of filtering. This software helps us by saying quality scores in the red, you don't want to use that at all. Quality scores in the yellow, perhaps you don't want to use it. Quality scores in green mean that 99.9 or 99.99% of our bases are A's, T's, G's, and C's we trust to be correct. We trust our data very, very much. So if we look across it, we can say, this DNA sequence for these Gila monster individuals is really, really good, and we think we can move forward with it. So imagine it would be like you're putting a puzzle together, but some of them have been gnawed on by your dog. You don't exactly know where they go, right? And they're kind of suspect. So you maybe leave those out till the last bit.
The equations help identify special DNA sequences and understand how similar tissues evolved and fit together. Once those pieces are matched, the Gila's genetic puzzle starts to take shape.
And then once we have the overlapping bits, then we want to know which parts are genes and which parts are not. And we do that by comparing to other species. So we do have what's called gene annotations. We know where a gene starts and ends in other species, and we use that to help us understand where they might start and end in Gila monsters.
However, they are still a very long way from seeing how each piece fits together.
It took multiple labs across continents to develop a reference genome for humans. So for humans, we have that picture, and then for each individual, we can look at their DNA relative to that picture and see where small differences are. And it helps us understand where mutations or differences are that give us different characteristics or susceptibility to disease. We'd love to do that for the Gila monster, but we don't have that picture, we don't have that reference. And so for this project, what we had to start off doing was make that picture. Putting those puzzles together is still really time consuming and very expensive, and not a lot of funding agencies want to support it. And so this project in particular has been entirely supported by my lab and by a crowd-funded project where it was people from the general public who were helping to support the reference genome of this species. And I think that's really fantastic. It means that it's going to take a little bit longer to put everything together.
By learning how Gila monsters evolved, we can understand their connection to other endangered species and predict how they might adapt to changing climate conditions.
So having genomic resources will help with conservation and with studying them. So they are protected in the state of Arizona. You can't own a Gila monster in the state of Arizona. You can own and breed them outside the state of Arizona. But in the state of Arizona, you're not allowed to touch them. Gila monsters coming in contact with humans are going to happen more often as we encroach on their habitat. So right now, we don't have a really great idea of how much genetic variation there exists across Gila monsters. So is there a very large effective population size? Are there many of them that are out there and hidden that we can't see? Or is there very little genetic variation across Gila monsters, and are they going to be really susceptible to things like climate change, humans building houses in their territories? And we won't know that until we can go out and sample their DNA and compare it to some sort of reference.
It's one example of how researching animals helps our own species adapt to future threats. After a long day, Tammy Borgard loves to unwind in her pool.
Just sitting and relaxing and floating and just the soothing of the water, because I have a metal piece you got to remember, so if I'm out in the heat, that metal is contracting the heat and that goes up into my bone. And then in the cold, the same way. It's getting onto that metal piece and it goes up into my bone. In October 2010, I got in a motorcycle accident. Somebody T-boned me, and I had angels around where I was at. They held my arteries until the medics came, and then we amputated, and the rest is history, basically just being in the hospital for about 2 months. So I came home, made it home for Christmas that year. Amputees wear sockets, and what it is your stump is in like a bucket so that you can walk, and all the pressure points are in between your muscles and your bones so that you can walk in that bucket, and they form it to you to walk as best as you can. So I was going back and forth trying to be able to fit, be able to do regular stuff in a socket, and it wasn't working, and I was always in pain. So that went on about 3.5 years. So I'm involved with an amputee support group at Health South. They introduced me to this doctor from Australia, and he says that they do implants into your bones, and that gets rid of your socket, and all you need is your prosthetic foot. So I took a leap of faith and went to Australia to get it done in April of 2014, and I have never walked better since my amputation. Not to say it's the perfect fix, but it's a lot better fix.
Scientists want to help amputees like Tammy avoid the discomfort of implants and attachments. That's why biologists at the Kusami lab are using genomic sequencing to uncover which regenerative genes come into play when lizards regrow their tails.
A lot of animals can regenerate, and so if we think about vertebrates or things that have a spinal cord, right, people usually use frogs or zebrafish or even salamanders as common model organisms for regeneration. However, the lizard is also really unique because if we think about the evolutionary history, reptiles, including lizards, are more closely related to mammals, which include humans, and so they have that capability to regenerate appendages and are more closely related to the humans than any other regeneration model itself. Humans can also regenerate the peripheral nerves. The issue is that, functionality isn't restored. And so luckily in these lizards at around 120 and 250 days, the nerves are also maturing, but also we begin to see that function is restored. So we're able to autotomize these lizards, and then we usually let them regenerate anywhere between 15 days all the way out to 250 days. And we'll collect them at certain time points. And so for our purposes, what we usually do is we'll take the tail and we'll fix it. That's just to maintain any of the connections and also preserve the tissue structure as well. And then doing that, we actually thinly section it into sections so that we can apply immunofluorescence. And so immunofluorescence is a technique where you can use antibodies which are specific to proteins, which we can use to identify certain tissue structures and see how they differ between the original and the regenerated tail. And then these antibodies are also connected to a secondary antibody that when hit with light will fluoresce and produce a different color spectrum. So that way we can label muscle red, we could label nerves green, but ultimately we could compare how the morphology of a regenerated tail compares to an original tail.
Their research focuses on a special type of cell known as satellite cells that are vital to the healing process.
Satellite cells are muscle stem cells. We all have them. Basically, all complex organisms have them, and they're really critical for muscle repair. And so when you've suffered an injury or if you have some kind of a muscle disease, or even after like a really good workout, these cells are able to wake up, sense the damage, and then go over there and basically repair whatever has been lost. And so they're very, very important for muscle repair. We can't really fix muscle without them. We primarily work with mouse, which is a good mammalian model, but they don't really regenerate very well. And so we moved on to lizards. It's difficult to study stem cells in the animal. There are a lot of different variables. And so what we do is we take those cells and we cultured them in an incubator so we can just study the stem cells and their behavior without all the other confounding variables. And so we would take the cells, grow them up, and then we can do all kinds of experiments with them. We can put them in a micromass culture, which is known to promote cartilage. We can look at what kinds of genes they're expressing. We can look at what kind of proteins they're expressing. So we can do all of this work without worrying about all the confounding variables that are in a person. And so that's what we set out to do with the lizards. And I believe we were the first to try to culture lizard satellite cells. And so it was a pretty difficult process because it turns out that all the things that work for mammalian cell culture did not work for lizard cell culture. And so we had to establish a whole new culture system. We had to figure out like what the optimal temperature was, and we had to figure out like what kind of media to use. And so this was really like the beginning of the process for us. So it's almost like having little pets that need to be fed and moved around and kept happy. Our main finding was that mouse satellite cells, and by the expansion, all mammalian satellite cells, are monopotent. So they can only become one thing. They're destined to become muscle, and that's all they can really do. But what we found is that lizard satellite cells, even though they behave the same, they look the same, they have this expanded ability. So they can not only become muscle, but they can also become cartilage. And so this might underlie some of the expanded regeneration ability that we see in the lizards. The hope is that we can take what we've learned from the lizard satellite cells and eventually, in the future, translate it to human work. Because the reality is that we share the same genes. So the pathways that the lizard satellite cells are activating are pathways that we have. They're very conserved. And so it's really just a matter of accessing that or tapping into that.
Being able to regrow bone and cartilage could help patients like Tammy move around more smoothly.
So my biggest challenge is always learning how to walk and making sure I'm walking correctly and I'm walking straight. One little adjustment off, I can screw up my back and be in back pain for a day or two. If I had grandkids, would I be able to chase after them, just being able to have that freedom to be secured on again, being normal, being able to do what your body's made to do. I'm always looking for new technology. Anything like my osseointegration implant, that's a leap of faith that I took that, is this gonna work? And if not, worse things could happen. So regarding the lizard stuff, that would be awesome. I would love to see it for the legs, the limbs, but I really see it for maybe like toes and fingers first of all starting off, and I think it'd be great.
Although their research is exciting, the scientists warn that translating lizard research into human healthcare is not as simple as tweaking a couple of genes here or there.
Obviously, it's not going to be a year or two that suddenly we have therapies to do regenerative medicine. We're not going to be able to just inject a gene or tweak a pathway and suddenly we can regenerate a full arm. But I think it is important that we understand, what's actually going on at the molecular level and the cellular level to kind of identify why lizards have this and have maintained this ability to regenerate appendages versus why humans can't. And if we can understand that difference, we can hopefully develop future therapies in order to take advantage of those things in humans and be able to direct the regeneration process as well.
I don't want to sensationalize it too much. I don't want to, say that we're going to be growing limbs tomorrow. But I think, we found that lizard satellite cells can become cartilage. And I think the next step is, well, why? What genes are they turning on and how are they doing what they can do? And then trying to apply that to humans.
In the meantime, Tammy is staying active with the limbs she still has.
Life goes on. There's so much to live for in my eyes. Mornings are the best part of the day for me. Being able to see the kids growing up or being able to see something beautiful. Anytime like the emotional part, you always have that, I don't want to get up, I have to put on my foot, I have to clean this, I have to do this, you have to do that. I probably struggle sometimes with that every morning, but by the time you get up and you start moving, it's a good feeling that I'm walking. Just look at the beautiful things to fight for. Don't settle. Just keep looking for what's going to work for you. How nice would it be to be normal, not have to have all these metal pieces on you? Technology is amazing, and if somebody can take that challenge and see if it'll work for them, I say go for it. If it works, I'd probably be one of the first ones to try it, to tell you the truth, if it would work.
I'm Professor Vanessa Ruiz, and thank you for watching Catalyst, our show about science. And because science always has new questions to answer, we'll be back soon with more stories.
Catalyst is supported by Knowledge Enterprise Development at Arizona State University, advancing entrepreneurship, innovation, discovery, and knowledge for the public good.
Subscribe to this channel to see more episodes of Catalyst on YouTube.